Hello, welcome to Biology for You. In this video, we are going to discuss central dogma. In the series of videos, we have been discussing different concepts in molecular biology. So, in this video, we are going to have the basic idea of the central dogma of the life or central dogma of the molecular biology. So, if you go by the name, the dogma, the word indicates the theory or the concept which held unquestionably, or the concept which have the undefended certainty. So that kind of central dogma was proposed by the famous scientist known as Francis Crick. So he postulated this concept in the year of 1958. So basically, central dogma of molecular biology or central dogma of life describes how the cell functionalities will be going on. That means at any point of time, as I am moving my hand, the cellular processes will be going on. So in order to move my hand from here to there must be different functions that need to be carried, like the muscle contraction must be carried in my hand tissue. So likewise, for doing any function by any kind of cell, that means dependent on the type of cell, different functions will be carried all over the time. So that will be carried by the information they are getting. That means the information resides the cell itself, guides the functions of the cell. So here basically, as we all know, a cell is the functional unit of the organism. So inside the cell there will be guidelines for its function. So here inside the nucleus there will be genetic material or the DNA. So mostly the genetic material is the DNA, which resides inside the nucleus for the case of eukaryotes, and it has the information regarding the cellular functions. That means at the functional level the proteins will be there. That means as we considered an example of moving a hand from one place to the other, we need to have muscle contraction. That means for the muscle contraction, some of the enzymes must be employed. So for that, all enzymes are nothing but the kind of proteins that have the different catalytic functions. So for any structural or catalytic function, all kinds of cellular functions will be carried by proteins. So these are the macromolecules which carries different functions in the cells. So these proteins will be spread all over the cell, but these proteins have the information to do some particular function. So that will be guided by the DNA which present inside the nucleus. So this DNA must have the information how to produce different proteins, how to regulate their functions. So that will be carried by the DNA which resides inside the nucleus. That means somehow it need to transfer this information to the functional level, that means to the proteins. So the DNA will be producing something called RNA, which is the intermediate between the DNA and the proteins. So in the central dogma, we are going to have the idea how DNA which resides inside the nucleus will decide the functions of the cell. So here basically there are at least 3 steps. First, primarily 3 steps in the central dogma. That means the DNA will produce RNA and RNA will produce proteins. So that is the baseline of the central dogma of the molecular biology. So proteins are at functional level and they carry different structural and functional processes of the cell. So here the information in the DNA also must have to maintain the information to the further generation. That means as it acts as a genetic material, it need to have the information for its own replication. So this DNA will be segregated to the further generations. So if the cell want to go for division and to produce daughter cells, the DNA complement must be properly segregated and equally partitioned into two of its daughters. So to segregate or partition its own DNA into all of its daughters, it need to replicate and make it into 2 copies at every replication event. So the DNA must have the information for replication of its own and for production of its RNA. So this RNA acts as an intermediate which has the information regarding the proteins, and the RNA will be smaller than the DNA, which can squeeze through the nuclear pores and goes into the cytoplasm where it need to produce some proteins, where the most of the proteins function will be carried. So this is the central the idea of the central dogma of the molecular biology. So coming to the processes in this central dogma, as we already discussed, DNA so from DNA to DNA, that means the first process is that DNA should replicate. That means it need to multiply its information so that the copies of it will be segregated to the further generations. So DNA must have the capacity to replicate itself into multiple copies. So that is known as replication. So first process in the central dogma is the replication. That means DNA should duplicate into its own complement. So that is the replication, by which DNA will be duplicated into other copy of the DNA. So the next one is DNA need to give information in the form of RNA. So here DNA cannot always give information directly to the proteins, but it will be doing this through the intermediation of the RNA. So RNA is the intermediate molecule which can squeeze, which can be produced inside the nucleus, that can be squeezed through these nuclear pores and coming out into the cytoplasm, where it can produce the proteins. So the second step is DNA need to pass information to the RNA. So that is known as transcription. So DNA will be giving information, passing information to the RNA. And the third process is these RNAs have information regarding proteins. They'll produce proteins in the process called translation. So this is the third process of the central dogma. So through the replication, DNA will produce another DNA, new copy of DNA, and through the transcription, DNA produce RNA, and this RNA will be going out, and that will translate it into proteins, different proteins. These proteins will monitor all cellular functions. So this is the central dogma of the molecular biology, and here basically all this information is in the form of codes. So that means DNA, as we already know, it is double helical structure which have many base pairs on it. So these base pairs of nucleotides, one nucleotide will be binding with other nucleotides. So these a chain of or polymer of nucleotides have the information. So on one strand of DNA, during the process of replication, the strands will be separated and each strand will act as a template to copy the other strand. So the other strand of the DNA will be formed by copying the nucleotide sequence present on the parent strand. So one strand acts as a parent to produce its own complementary strand, which is new strand, to produce a complete new strand, and two of such new strands will be produced from one DNA in the process called replication. So there are enzymes which helpful to initiate the replication and will copy all the DNA present in the chromosome, and likewise the whole genetic material will be copied or duplicated into two. So that is what the replication is. So DNA in the form of nucleotide sequence will copy its own information to produce another copy of DNA in the case of replication. So coming to the next step, that is transcription. So in which the DNA, again the two copies of the two strands of the DNA will be separated, and here the nucleotide sequence will be copied in the form of RNA, not in the form of DNA, by employing process called transcription. So there are some enzymes. These transcription enzymes will copy the nucleotide present at small stretch of DNA, and these will be copied into its mRNA. So here the nucleotides are slightly changed, and the portion of DNA which is only meant for the production of a certain protein will be copied into the form of mRNA, because not all the time all genes are needed to all genes are all not all proteins are needed all over the time. So only the proteins which are needed for the point of time are produced in the form of mRNA or copied in the form of mRNA. So these mRNA will carry information regarding certain proteins which are needed for the point of the time for a cell. So these kind of process is known as transcription, in which the DNA will be producing this mRNA. So along with mRNA there are other RNAs, tRNAs, rRNAs are also be produced, and these are also helpful for the translation of the RNA into the proteins, which is the third step of the central dogma. So coming to that step, the mRNA which is produced inside the nucleus will be squeezed through the nuclear pore most of the times, and it will come out into the cytoplasm where the proteins are produced, and these mRNA carry information in the form of nucleotide sequence. So basically these nucleotides, the code is in the form of triplets. That means 3 nucleotides together make a code. So genetic code is always in the form of triplets. So in the triplet genetic code, it codes for the proteins. So basically proteins are the chain or polymer of the amino acids. So in order to produce proteins, we need to make chain of amino acids or the polymer of amino acids. So each triplet code, that means 3 nucleotides, each of 3 nucleotides or the triplet codes for the amino acid, each amino acid of the protein. So likewise, a chain of amino acids will be produced from the mRNA during the process known as translation. Basically, this is carried on the part called ribosome. So on the ribosome, the proteins will be translated and using the help of tRNA. And ribosomes have their own RNA called rRNA, and both of them are helpful to produce proteins. This tRNAs actually which brings the amino acids to the site of the protein synthesis. So they carry amino acids to the site of protein synthesis, and these rRNAs, which are the part of the ribosomes, which are the missionaries of the protein synthesis. So these ribosomes are the missionaries of the protein synthesis, and these rRNAs are part of it, and by which the mRNA which bind to these ribosomes will be translated into the proteins. Here one amino acid will bind to the next adjacent amino acid, which will be guided based on the information on the mRNA. So likewise, a chain of amino acids will be obtained, which are bind together by the peptide bonds. So this polymer of amino acid will be produced, which are stitched with the amino peptide bonds, is known as protein, and the whole process is known as translation. So mRNA, which came out of the nucleus, will be coding for the amino acid sequence of the proteins, and all over the process will be carried on the machinery of ribosomes, on which mRNA will be binded, and tRNAs will bring the amino acids at every point of translation. So this is all the idea of the translation. And coming to these are the basic processes in the central dogma of the molecular biology. So coming to the other concept, which is reverse central dogma. That means basically this is unidirectional process. At first we thought the central dogma is unidirectional, so the information cannot go back. That means the information in DNA will be moving forward in the form of RNA and in the form of proteins. So the protein cannot give information to the RNA. That means the process is unidirectional. But after the discovery of the reverse transcriptase enzyme and mostly the discoveries of David Baltimore, the scientist who proposed the reverse central dogma, in which the RNA sometimes in some of the viruses like tumor viruses will give information to the DNA. That means here in the tumor viruses, the genetic material is RNA but not the DNA. So in those viruses, the RNA will be copied into the complementary DNA or cDNA. The process is known as reverse transcription. So, in order to influence its host, the RNA present in the virus will be copied into its cDNA, and the cDNA will be again producing RNA, and that RNA will be again producing the proteins for the viruses. So, in the viral infection, the reverse transcription will happen. So, in the cancer or tumor causing viruses, this reverse transcriptase enzyme will be there, which can reverse transcribe the RNA to produce its complementary DNA. So this is the fourth step in the central dogma or the reverse central dogma, that in which the DNA the RNA will be reverted in the form of DNA. So this is all about the idea of the central dogma of the molecular biology or the central dogma of the life. So if you really like the video, please share and subscribe to my channel.